We are now going to take a look at another scenario that is an exception to the rule. In this instance, we will take a look at genes that are located together on the same chromosome. Because they are located on the same chromosome, they then violate Mendel's law of independent assortment. In all of the previous cases that we've seen and all the previous crosses we have done, we've assumed that Mendel's law of independent assortment has held true. In essence, that means that the genes that we were looking at were present on different chromosomes. However, this is not always the case. One of the tests that we can do to see if genes are linked is to do a test cross with a dihybrid individual. In this instance, one of the parents would be a full recessive individual. This is the definition of a test cross, whereas the other parent in the cross would be a heterozygous individual. There are two types of offsprings that can result from this test cross: parentals and recombinants. Parentals offspring will end up showing the combination of traits that we see in the parental generation. In this case, they would exhibit the dominant phenotype and the full recessive phenotype. Hence, those are the parents used in this cross. The recombinant offspring will show combinations that are not seen in the parental generation. They would be dominant for one trait and recessive for another, and vice versa. We can see from the diagram below how we can refer to our offspring as parental or recombinants. Note that when we are doing a test cross using a full recessive individual, there's only one type of gamete that can result from that parent, and that would be, in this case, little y, little r. In a heterozygous individual, we have four different gamete possibilities: big Y, big R, big Y, little y, big R, and little y, little r. We can see in the cross below that when we look at the Punnett square that results from those particular gametes, we have four different offspring possibilities: big Y, big R, little r, and this would be considered a parental. Note the same genotype would have resulted from the parent that we used in this cross. The second type of offspring we would get is little y, little r, little r. This is also classified as a parental offspring. We then have offspring that have big Y, little r, little r. Note that they would exhibit the dominant yellow phenotype in this example, but they would be wrinkled, which is the recessive trait. This would be an example of a recombinant offspring. Conversely, we would have the final type of offspring being recessive, little y, and then exhibiting the dominant round. However, the genotype would be big R, little r. This would also be considered a recombinant offspring. Here is another example of studying gene linkage in flies. In this instance, we have the parental generation being homozygous for the wild-type alleles. In this instance, the genetic makeup would be B plus B plus, designating a gray body, and VG plus VG plus, designating normal wings. This would be crossed with a double mutant that is a full recessive individual. This would also be considered a test cross because we have a full recessive individual as a parent. In this instance, the genetic makeup of the mutant would be BB, signifying black body, and VG signifying vestigial wings. The result from this cross would be creating a dihybrid or heterozygous individual with a genetic makeup of B plus B, VG plus VG. We would then take this heterozygous individual and perform another test cross by crossing it with a double mutant once again. The genetic makeup of that double mutant would be B, B, VG, and it would have a black body and vestigial wings. Now let's take a look at the gametes that would be produced by these two individuals. Once again, from the mutant male, we would only have one gamete possibility, and that would be BVG, both mutant alleles. In the eggs, however, we have four different gamete possibilities. One of those possibilities would be both wild-type alleles, B plus, VG plus. We would also have a B VG. We would also have a B plus VG. And finally, a gamete with B VG plus. If we take a look at the resultant offspring that would result from fusion of these eggs and sperms, we can see what the offspring would be genetically and phenotypically in the diagram below. We would have wild-type gray and normal flies that would have the genotype B plus B, VG plus VG. Note that this would be considered to be a parental, because this is the same genetic makeup as our heterozygous individual in this cross. We would also have black vestigial flies with a genetic makeup of BB, VG, VG. This would also be considered a parental. This is the same genetic makeup as our double mutant male used in this example. Together, those two fly groupings of offspring would be considered the parental offspring. Continuing on with the cross, we would have some of the offspring having the genetic makeup of B plus B, VG, VG. These individuals would exhibit the gray or wild-type body coloration. However, they would have vestigial or mutant wings. We would also have individuals that have BB, the black mutant body type, along with VG plus VG normal wings. These two offspring types are considered to be the recombinant offspring. If we consider what our expected ratio would be if these traits were not linked on the same chromosome and if they followed the law of independent assortment, we would note that all of these offspring potentials would be equally likely, such that they would be present in the offspring population in a 1-to-1 to 1-to-1 ratio. If these genes were linked on the same chromosome, however, we would have more parentals than recombinants. If we take a look at the ratio below, we can see that our observed ratio in this cross would be 965 individuals that have wild-type conditions, a gray body and normal wings. We would also have 944 individuals that have black body and vestigial wings. These would be considered the two parental possibilities. We also see that the results indicate that we have 206 flies that have gray bodies and vestigial wings, and 185 of our offspring have black bodies and normal wings. We can see that there are many more parental offspring than recombinants. This indicates that these genes are in fact linked on the same chromosome. To understand if we have genes that are linked on the same chromosome, it is important to know what we would get from our expected Mendelian ratios. Remember, our expected Mendelian ratios are what would happen if these alleles separated themselves independently and followed the law of independent assortment. If they do not follow this, we will see differences in the offspring ratios, which indicate that these genes may be linked. Another important thing to understand is that if these genes are linked together on the chromosome, however, if they are farther apart on the chromosome, we know that we can have crossing over events that occur during prophase. This crossing over event can act to unlink these genes through genetic recombination. Genes that are located on separate chromosomes always undergo genetic recombination because they are never linked together. As a result, they follow the law of independent assortment and would also follow the expected Mendelian ratios. In order to test for genetic linkage, we can ask ourselves, do these two genes assort independently? And do we get our expected Mendelian ratios? Some of these expected Mendelian ratios would be a 9-to-3 to 3-to-1 ratio for a true heterozygous cross. Remember, the only way that we get a 9-to-3 to 3-to-1 ratio is if we are crossing two heterozygous individuals. That 9-to-3 to 3-to-1 ratio exhibits that 9 of our 16 offspring would exhibit the dominant trait. 3 would be dominant for one trait and recessive for another. The other 3 would be recessive for one trait and dominant for the other. And one individual would be a full recessive. We can also test against our expected Mendelian ratio of 1-to-1 to 1-to-1. However, this is only when you are doing a test cross with a heterozygous individual. We can also use this principle to test for genetic linkage if we understand that we are getting more parentals than recombinants, and that this ratio would differ from our expected Mendelian ratio that would occur if the genes were sorting themselves independently as if they are on two separate chromosomes. Simply stated, the expected ratio that you have will differ depending on the actual parents that you have in the cross. By understanding how many parentals we have versus recombinants, we can determine what the recombination frequency is. This is also known as the RF value. This can be calculated by taking the number of recombinant offspring divided by the total number of offspring in the cross. The recombination frequency, or RF value, is an indication of how far apart or how close together these genes are on the same chromosome. RF actually translates into map units. Note that as the distance between two genes increases, the probability that they will unlink also increases. This is because the further apart these genes are on the same chromosome, the more likely they are to separate out when they have a crossover event. In general, if your RF value is greater than 50%, these genes are considered to be far apart, are considered to be unlinked, and assort themselves independently. Thus, they would follow expected Mendelian ratios in your cross. However, if your RF value falls less than 50%, we would determine that these genes are close together and are in fact linked on the same chromosome. The diagram below gives you an idea of the different genes and how they are linked together. This is a typical chromosome map. We can see that if we are looking at two traits, such as short aristae, which is short antennae, versus long antennae, and we also look at the trait for maroon versus red eyes, we would see that these two traits are located very close together on the same chromosome. In fact, they are only 16.5 map units apart. This indicates that these genes would not be assorting themselves independently, and you would in fact, in this type of cross, get more parentals than recombinants. However, if you were to be looking at, say, the short aristae trait, and be looking at it with vestigial versus normal wings, we would see that those units are in fact 67 map units apart. Because these two genes are located so far apart, greater than 50%, on the same chromosome, if we were doing crosses with these traits, there is a high likelihood that they would undergo genetic recombination, and that means that our expected ratios would closely match our observed ratios. In this instance, between these two genes, they are more likely to jump on different chromosomes and behave as if they're assorting independently. Similarly, if we were to look at vestigial wings and let's say cinnabar eyes, those two traits are located and genes are located very close together on the chromosome. If we were to be specifically looking at those traits and do genetic crosses with those traits in mind, we would be very likely to see many more parentals versus recombinants in that instance. We can also have various types of mutations in chromosome number. Aneuploidy refers to any alteration in chromosome number. Many conditions of aneuploidy are due to a non-disjunction event. This is when the chromosomes do not separate out properly during meiosis. Non-disjunction events can commonly occur in meiosis 1, when the homologous pairs of chromosomes or tetrads are separating, or they can occur in meiosis 2, when separating out the sister chromatids. You can also have a total non-disjunction event, which means the complete failure to separate out all of the chromosomes involved in producing gametes. This would lead to the condition of polyploidy, having two or more sets of chromosomes. This can occur in some plants, insects, and fungi with non-detrimental effects, meaning that the organisms can survive and reproduce. Most aneuploidies, however, lead to death in humans. The result of non-disjunction event is typically one gamete will be missing a chromosome, where the other one has an extra chromosome. Remember, it is very important to have haploid gametes, and so when this does occur, This will commonly lead to a miscarriage. If either of these gametes end up fusing with a normal gametes, we will have either a trisomy or monosomy condition. Trisomy would be resultant from the gamete that carries an extra chromosome, and a monosomy would be from the gamete that was lacking one. If we take a look at the diagram to the lower right, we can see what would occur in the gametes from a non-disjunction event in meiosis I and meiosis II. In a non-disjunction for meiosis I, we can see that one of the tetrads fails to separate out properly. As a result, one of the two initial daughter cells produced from the pregametic cell would have one of two conditions. One cell would have the extra chromosomes, whereas the other one would be missing. As a result, we can see that all four gametes produced from a non-disjunction event in meiosis I would have a genetic abnormality. Half of the gametes would be considered N+1 and carry an extra chromosome, whereas the other half of the gametes would be N-1 and would be missing a chromosome. Any of the gametes that are N+1 would form a trisomy condition in an embryo, whereas any of the offspring that result from the gametes with an N-1 would be considered a monosomy condition. If we take a look at what happens if we have a non-disjunction event in meiosis II, we can see that now our non-disjunction event happens in one of the two cells that resulted from meiosis I. In this instance, it is the cell to the left that is having the non-disjunction event. As a result, we can see that any of the gametes produced from that specific daughter cell would have a genetic abnormality. Half of the gametes would have N+1 and the other half would be N-1. If we take a look at the gametes that would result from the second daughter cell that does not have a non-disjunction event, we can see that they would have a normal haploid state of N. In this instance, we have the chance to have normal individuals if the N gametes end up being fertilized. However, if we have one of the N+1 or N-1 gametes that end up fertilizing and forming the embryo, we would once again have a trisomy and monosomy condition. Most monosomies and trisomies are considered to be lethal very early in development and typically lead to miscarriage. However, one of these conditions, specifically trisomy 21, is not considered to be lethal. However, most Down syndrome individuals are considered to be sterile and not able to reproduce. However, this is not always the case. In trisomy 21, the individuals have 3 copies of chromosome 21, which we can see in the karyotype below. If you orient yourself, you can see that in the bottom row of chromosomes, we can see that chromosome 21 does indeed have 3 copies. This chart below shows you the various disorders in chromosome number when you have aneuploidy in the sex chromosomes. There are various combinations that this can take. We can have individuals that have 2 X chromosomes along with a Y, also known as Klinefelter syndrome. Some individuals may only have an X chromosome, meaning that they are missing the additional X or Y, also known as Turner syndrome. We can also have individuals that are XO or XY, individuals that have an extra Y chromosome, individuals that have 3 X chromosomes, and individuals that are XX and XY. Please take a minute to refer to the chart below to see the different characteristics of individuals with aneuploidy in the sex chromosomes. In addition to alterations in chromosome number, sometimes the chromosome structure itself can be altered. Chromosomes, when they are being replicated, duplicated, and separated, can sometimes get breaks and DNA damage. This can lead to various types of conditions, such as a chromosome deletion when part of a chromosome ends up being lost. There can be duplications where a sequence of DNA is accidentally repeated. Sometimes this sequence of DNA can be inserted backwards into the DNA or into the chromosome, which would lead to an inversion mutation. At times, there can be a translocation mutation. This occurs when part of a chromosome breaks off and attaches to another chromosome. This can be a reciprocal translocation if you have a mutual double switch of chromosome fragments. This would be equivalent to having a crossover event between 2 non-homologous chromosomes. We can see here a common condition is to have a reciprocal translocation between chromosome 9 and chromosome 22. In this instance here, when we have a crossover event, we would see that our normal chromosome 9 would now have a small portion of chromosome 22 present on it. In our chromosome 22, we would note that it was particularly shorter and would also contain a portion of chromosome 9. This is typically called the Philadelphia chromosome. This can be easily identified in a typical karyotype. So it is very important to maintain proper chromosome structure and proper chromosome number for normal embryo and human development.